presentation.
The bottom left corner says share sound.
So I'm Jen Roggenbuck, and I am a genetic counselor, associate professor at The Ohio State University. And I'm so pleased to be here with you this morning to give you a hopefully friendly introduction and overview about titinopathies, which is, of course, what brings us all to this particular room today. And what I hope to do is give you a little bit of background about the titin gene, the conditions that can result when there are changes in the titin gene, and then also review some basic genetics, because all of this can get pretty confusing as we're getting genetic test results back and thinking about what those results mean for you and your family members. So my hope is that this little guide that I've put together will set the table with some basic terms and concepts, and hopefully there will also be time for questions. And I think as far as my time, I'm limited to 20 minutes. So if I get close to that, please let me know, because I have a feeling that this could go a little bit longer. Yes. Okay. Okay. So can everyone at home and around the world hear me? Yes, okay, wonderful. We're special welcome to our virtual attendees, especially the international ones. We're so excited to have such good representation at this session this morning. So titinopathies are genetic conditions which affect muscle. And this includes muscles used in movement, so moving the body. This is what we call skeletal muscles in the medical terminology. The heart is also a muscle, and that's the cardiac muscle that pumps blood throughout the body. And then in some titinopathies, the muscle that's used in breathing can be affected. So that's the diaphragm. But all of these muscles can be affected to a variable degree and in variable combinations in the titinopathies. Titinopathies tend to be named for the symptoms that they cause. And this can lead to some, I hope it's okay that I was putting this talk together and I wanted to spice it up with some human interest. So I really thank you for posting that beautiful picture, because when I saw that picture, I was like, I have to show that picture. Anyway, I just love it. But titinopathies are named for the symptoms that they can cause in the body. And this can lead to some very long, tongue-twisting names. But one of the more common titinopathies is titin-related dilated cardiomyopathy. So this is a condition where the heart muscle gets affected and doesn't pump as well. Tibial muscular dystrophy is one of the first skeletal muscle titinopathies that was discovered. Hereditary myopathy with early respiratory failure is one of the titin conditions that can affect the diaphragm quite prominently. So that is one of the distinguishing features of that condition. We also have some other cardiac conditions that are newly associated with the titin gene. And then there is a dominant cardioskeletal titinopathy that I added to this list kind of at the end, because that's a condition that I believe is an emerging condition that I personally am studying. So I'd be happy to tell you more about that later. But this is a mild adult, typically adult or late childhood onset condition that can affect skeletal muscle and cardiac muscle. So all of those conditions that are listed on the left are conditions that can be passed from generation to generation. So we can see parents affected, children affected, though there might be quite a bit of variability in those symptoms. So not everyone in the same family across the generations will necessarily show the same symptoms. Now on the other side, on the right-hand column, we have recessive titinopathies. So these are conditions that typically can affect siblings, but not across the generations. So we don't see affected parents and children with recessive titinopathies. So this includes centronuclear myopathy. This particular condition is named for the appearance of the muscle on a muscle biopsy. So this is not necessarily a symptom, but it's more the appearance of the muscle tissue. We also have titin-related distal myopathy, That's a recessive, usually young adult onset condition where there can be weakness in the hands and feet. And then there are some additional congenital conditions that are really too numerous to name here, but that includes early onset myopathy with fatal cardiomyopathy, as well as a variety of arthrogryposis conditions. And arthrogryposis means contractures. And we can see babies born with contractures in their joints with their hands folded and their limbs with limited mobility. So there's quite a range in the symptoms, but what's common to all of these conditions is weakness in the muscle that leads to skeletal muscle weakness, heart failure, or in some cases, congenital differences with both muscle, breathing, and the ability to move. So what is titin? So titin is a protein, and believe it or not, it is the largest protein in the body. It is huge. And that structure that it lends muscle is what allows our muscles, which are relatively small, to generate immense force. So it really is miraculous what our muscles can do as human beings. And this titin protein is wound up with other muscle proteins, including actin and myosin, in these very tight springy structures that exist in our muscle fibers that allow our muscle fibers to expand and contract at will, and do so repeatedly over the long course of our life, both in our skeletal muscles and in our heart. Now, the other amazing thing about titin is that there can be slightly different versions made from the genetic code, and I'll get to that in a minute. But these different versions are called isoforms, and there are slightly different isoforms in the heart versus in the skeletal muscle, and also at different times in human development. So there's some titin isoforms that are present during fetal life that are not present in adult life. So titinopathies are caused by harmful changes in the titin gene. So the foundation of genetics is that every gene that we have is really a code that tells the body how to make a protein. And our body is completely made up of proteins, and even things like enzymes that are facilitating chemical reactions in our body are also proteins. So our bodies are completely made up of these proteins, and each protein has its own genetic code, which is like a recipe card that the body reads in order to manufacture that protein. So you can see how critical it is that that information is stored in a safe and accessible way, and that's what our DNA does for our bodies. However, if there are harmful changes in that genetic code, that can prevent the body from reading the genetic code correctly, resulting in an incomplete or a misshapen protein that doesn't work quite as well as it should. Or in some cases, the protein might be completely absent. And in the case of titin, this ultimately prevents the muscle from working correctly. And these genetic changes are usually inherited from parents and can be passed on to children. Now, the risk of a child being affected really varies depending on the condition and how that condition runs in the family. And I hope to explain a little bit more about that. So in order to do that, it's helpful to review some basic biology. And if you guys remember high school biology or even college biology, you might have learned about how exactly genes encode proteins. So on the top bar here, we've got our DNA strand, which is a double-stranded molecule. And those blocks that you see are actually the parts of the gene that are important. These are the parts that actually encode for amino acids that ultimately get put together into the protein. And in between those blocks are these stretches of blank areas, which is also DNA, but it's actually DNA that doesn't matter. It's like spacers between beads on a string. And when the body wants to make a protein, it makes a temporary copy of the gene, and that temporary copy is called RNA. And that RNA actually copies the whole length of the gene, including that spacer DNA that's in between the important parts. Then that copy leaves the nucleus where the genes are housed and goes out into the cytoplasm where the actual stuff happens and proteins are made. So then the RNA undergoes a special snipping process called splicing to get rid of that junk spacer DNA and put all of the sense together in these continuous blocks. And then that finished RNA product is what the body uses to actually read the code and produce the protein. And that happens with the ribosomes out in the cytoplasm. And the fascinating, amazing thing is that these exons, those blocks, can be put together in different ways and different sequences to create different versions of those proteins, those different isoforms that I was telling you about. So this is really miraculous. But the fact that it's so complex means that there are a lot of different places where there can be glitches and things can go a little bit differently. Sure, yeah. Yeah.
I get the DNA, right?
Uh-huh.
It stays there and then it makes a copy with the RNA.
Yeah.
And then what happens to the RNA copy? Does it
It
just
gets,
dissolve? Like it disintegrates?
well, first it ideally goes to the ribosome, and the ribosome is a structure that brings the amino acids together and builds them one by one together to make the protein strand. And then once the protein strand gets made, it gets folded up to its additional structure and then gets incorporated into the muscle, hopefully. So but after that happens, the mRNA at some point does get degraded. It can also get degraded before it gets a chance to do anything, and that's one of the things that can go wrong in genetic conditions, is that, you know, the RNA that gets made, maybe it's missing something, or it's not prepared completely, and it gets degraded. The body says, wait a minute, this is an abnormal thing that shouldn't be floating around in the cell, and we're going to break this down and look for some other RNA. Thank you for the question. It's great to get some interaction here. So moving on to, I remember I promised to explain a little bit more about dominant and recessive. We all have two copies of every gene, one that we get from our mom and one that we get from our dad. And in dominant conditions, only one of those genes needs to be changed in order for symptoms to manifest. So that means that each time an affected person has a child, there's a 50% chance that that gene would be passed on and a 50% chance that the child would be affected. So we can see what we call vertical transmission in family, parent to child, parent to child. Recessive means that having one copy isn't harmful for that person, because you can then just go to your backup copy of the gene and make sufficient protein. However, if you meet up with someone and have children with another person who also is a carrier of a recessive genetic change, if you both pass on that change to the child, then that child kind of gets a double dose of that change and can be affected with the condition. So the reason this is important is that we have titinopathies that show dominant patterns of transmission and ones that show recessive. And the confusing thing is we're not always 100% sure which it is. And that's important for families to know, is that not even the doctors and geneticists and genetic counselors necessarily have a complete understanding of all this. And that's a really important take-home message, is that we are all learning together. And it's very important to ask questions, document what you know, and remember that we're all human and we're all learning together. As I mentioned, different combinations of these genetic changes can cause different titinopathies, and it can get quite complex because there's titin being so large, there's a lot of different things that can go wrong with it. There can be a premature stop, such that the body stops reading the mRNA before it should. There can be one amino acid swapped out for another. There can be a splice site change, and that is when the body is snipping out those spacer parts of the DNA. There can be some parts of the gene that are skipped over or doubled when that mRNA is made. So there's a lot of different things that can happen, and this is another reason for the high complexity of Titin genetics. That is important to remember too. And I don't know if I'm not able to advance my slide further. I think I'm about out of time. Oh, okay, all right. So for some reason, I'm not able to advance my slides. One of the things that I sometimes help people with is, okay, yeah, so one of the things that I sometimes help people with is what I call genetic report literacy, because it is so confusing to look at genetic test reports. And when I say it's confusing, it is confusing for everybody. It is confusing for the doctors, it is confusing for the genetic counselors sometimes, and it is certainly confusing for families. So I wanted to take a few minutes to walk through the highlights of a genetic testing report and impart what is the key information that you need to know about your genetic test results. The first thing is that it is really important to get a copy of it. And I cannot emphasize that enough. And I'm always telling families and doctors to share the genetic testing report, because it contains so much information that you really cannot get anywhere else. So unfortunately, there's not necessarily a tradition in medicine of doctors sharing lab reports directly with patients. But I do believe there's an increasing trend towards that. And sometimes you can even view them with patient portals like MyChart and things like that. Although genetic test reports tend to be send-outs that actually get run at a different place and then sent back to the hospital and maybe scanned into the medical record in any number of different places, so they can be really hard to find. But anytime you have a genetic test, be sure you get a copy of the report. And if you've had one in the past, you can go back and try and get one. But just for the sake of clarity, I've not shown the entire report, but what's shown here is the results. And okay, so up here it's saying result carrier. So what that means is that it implies that this individual has one copy of a gene that's recessive. And it turns out that in this particular case, it's a little bit more complicated than that, but good lab reports will give some type of summary about the findings. So here it says, one pathogenic variant was identified in Titin. Titin is associated with a spectrum of autosomal dominant and recessive cardiac and neuromuscular conditions. So that basically summarizes what I just told you this morning about how the conditions can be dominant, they can be recessive, they can be neuromuscular, meaning skeletal muscle, or they can be heart. And then it says additional variants of uncertain significance identified. Wow, okay, that's a lot to take in. But here's the table where the actual genetic findings are shown. So this is really important to look at and understand, because this contains the key information about what genetic variants were identified. So most genetic test reports will have some version of this table, no matter what country you're in or what kind of commercial or research lab. Hopefully all this information is entailed. So here we have gene. That means the gene where the finding was found. So variant. Variant is a term that in the old days used to be called mutation. But as a field, we are no longer using that, because that implies a level of certainty about harm that we don't necessarily always have. So variant simply means there's a difference compared to the reference genetic code. So what we think is normal, but we don't always actually know what's normal or not. And that's why we chose a softer word, variant, to describe a finding that might be different from what most people have. So this C dot here, this stands for cDNA. So this is the actual change in the DNA sequence. DNA is like an alphabet that has 4 letters, and the sequence of those different letters is what imparts the genetic code. So here, what we're seeing is at this particular spot, at this milepost, instead of a G, we have an A. There is one tiny misspelling in this huge protein. You would think that would not really matter, because it's just this tiny change. But then it says splice acceptor. What does that mean? So that actually is telling us what they believe this genetic change causes in terms of how that gene is read and processed. So what this means is that this tiny little genetic change is hitting one of those spots that the body snips when we go from the precursor RNA and we snip out the junk DNA to create our whole complete RNA that has the exons. So this is what we call a splice site variant. And it's fairly common in Titin to have splice site variants, and more common than other genes just because of the high complexity of all the 360 some exons that we have in Titin. Now, there could also be other information here if the mutation were a different kind where an amino acid was swapped out, it would say that. Zygosity means was it present in one copy or two? And then this over here is really important too, variant classification. This means what is the interpretation of does this variant cause harm? So this says pathogenic. That is indicating that it's believed to be harmful. According to the American College of Medical Genetics, there's actually 5 categories that a variant can be classified in. This is the most certainty for harm. Now, under here, we have another variant of uncertain significance, and that is also common in genetic testing, particularly when we're testing multiple genes at a time. Now, if you have a variant of uncertain significance, it is truly uncertain, and you need to work with a doctor who knows genetics or a genetic counselor to learn more about it. Now, I just wanted to point out that these genetic test reports typically contain a lot of detail, and you need to go through all 6 or 7 pages to really glean all the information. Here the laboratory is actually giving us some really nice background about this particular variant. So it's saying that it affects the splice acceptor site at intron 242, that's in the I band. It is expected to disrupt RNA splicing and likely results in a truncated or disrupted Titin protein. And here it's telling us that it's present at low frequency, very, very low frequency in reference population databases. And then it goes on and tells a little bit more about other cases where this variant has been seen. Another thing the test reports do is they tend to list all of the conditions that can be caused by a particular gene. And I've seen a lot of confusion with doctors mislabeling patients with a different condition that happens to be listed on the report merely to provide background. So just because a condition is listed somewhere on your report, that doesn't mean that's the actual condition that you have. So you really need to be careful and read the report carefully. And if you don't have someone who's on your team that is confident, reach out and try to find someone who is. And getting your test report and reading it carefully is step number 1. And it's okay to feel overwhelmed and confused because we all do when it comes to this gene. So I think I'm about out of time, but I look forward to your questions. And I'm so excited to hear about the ways that we can learn together this morning.
Thank you, Jennifer.
Okay.